Okay, so as people are trickling in to our webinar today, I want to welcome everyone to the Meta-eukomics webinar. We're continuing to host these about every other month, where we invite two speakers from our community. They each give about a 20-minute talk, and there's time for more targeted questions, and then we will move into more of a larger discussion if that kind of takes us there. Yeah, so I wanted to welcome everybody. And my name is Sarah Hu. I'm a assistant professor at Texas A&M. This is, I'm kind of I'm our working group for Meta-eukomics, which is funded through the OCB group. It includes Natalie Cohen, Harriet Alexander, and Adrian Marchetti. And a lot of them are here today. And we're excited that you can all join us. So today we've invited two speakers, Dr. Sonya Dyhrman and Dr. Lucia Campese, and each of them is going to talk about their work that uses meta-omics, which is kind of the core theme in these webinars. Today, I'm excited because they're both kind of set at these really historical oceanographic sites that have been monitored over a long time. And I think that's something that I've been thinking a lot about recently, of like how these important sites over the long term contribute a lot to our work, and how they kind of persist and outlast a lot of other things that might be happening in the world. And so I think it's nice to consider how those large group projects with some of the neat products that come from that. So I'm excited to hear from both of them. First up, we're going to hear from Dr. Sonya Dyhrman. She's a professor at Lamont-Doherty Earth Observatory, part of Columbia University, and she runs the microbial oceanography group there. And today we're going to hear about the physiological ecology of the microeukaryotes in the North Pacific subtropical gyre.
Thank you, Sarah. I'm going to just take a moment to share my slides here. Okay. Well, the title of my talk today is, hold on, I just need to put this down so I can see it. Okay. The title of my talk today is Learning the Rules of the System, Tracking Microeukaryote Physiological Ecology in the North Pacific Subtropical Gyre. And I actually wanted to start with something that I think this audience can all appreciate, and that's we're facing some grand challenges. One of the challenges that I think about a lot is that phytoplankton and, of course, other microbes underpin ocean ecosystem functions and the Earth's biogeochemical cycles, but we lack predictive power on a changing planet. There are all of these nuanced, beautiful interactions between phytoplankton and other microbes, between the chemistry and the phytoplankton, and the baseline is changing under us. This presents a lot of challenges, but it also keeps things exciting, and I would say it presents opportunities as well. And in fact, a confluence of opportunity and uh, need in terms of identifying the rules that govern ecosystems' functions and their sensitivities in a changing ocean. And when we think about meta-eukomics, it's a powerful time. New methods, workflows, sequencing technologies have expanded dramatically even in the last 10 years. And this is really opening up our ability to ask and answer questions around this grand challenge. So today I'll highlight how we are trying to learn the rules of the system by using metatranscriptomes to reconstruct, simply stated, who's there and what they're doing when it comes to the larger, I would say, nano size class and above eukaryotic phytoplankton. And we're doing that by visualizing the eukaryotic phytoplankton with these metatranscriptomic tools and understanding what they're doing under different kinds of forcing. The system and the work that I'm talking about today is all set in the North Pacific subtropical gyre in the surface waters. This is arguably the largest biome on the planet, and we've been focused on drivers of the eukaryotic phytoplankton community structure and function in this system. Now, when you think of this vast oligotrophic system, you maybe don't think about the larger eukaryotic phytoplankton. This system, of course, is dominated by picoeukaryotes and the picocyanobacteria like Prochlorococcus, and they just really teem in those surface waters. But the euks are important too. So the eukaryotic larger phytoplankton can form large aperiodic blooms that have then concomitant effects on carbon cycling and food webs. There's a summer export pulse linked to diatoms and coccolithophores, typically every summer. And in fact, these larger, again, nano and micro-sized phytoplankton, when they grow to high enough concentrations, this can influence the biological pump and carbon export. So let's just drill down into thinking about this system a little bit more. I'm going to call these larger eukaryotic phytoplankton keystone because they're typically dilute, but they aperiodically play a keystone critical role in the community structure function relationships, particularly around something as important as carbon cycling. So I have here two scenarios, one where you have the typical oligotrophic pico-sized class phytoplankton on the left with potentially low levels of nutrients flowing in and different forcings and low export production. And that can be contrasted where a system maybe seasonally or when you have a bloom, you shift to higher export production, a stronger biological plump, and that may be driven by different kinds of forcing, such as nutrient input through vertical mixing. One of the uh, graphics that I've found inspiration in thinking of this push-pull between these two different example states in the system is this graphic of ecosystem drivers, where you can track that little ball as it goes back and forth on this kind of seesaw, and you could envision maybe nutrient inputs or some other type of forcing driving the system to that stronger biological pump and that greater export production. And then maybe it reverts back again when that forcing is gone. So again, I'm going to call these keystone phytoplankton for their role in these larger scale ecosystem processes. And over the last roughly 10 years, my group has been thinking about what are the rules that govern structure function relationships for these keystone phytoplankton in the NPSG. And we've been able to take advantage of different kinds of experiments and observations to try to understand this system. This includes monthly time series, diel time series, looking at eddy-driven forcing, looking at natural disturbances. That's a picture of the lava flow from the Kilauea eruption, and using deckboard incubation experiments to really push and pull the system in different ways. And I'm actually excited to report that we're in the process of finishing up a monthly time series observation of euc metatranscriptomes from the surface waters that was collected on hot cruises. This spans almost 4 years, and it's going to be a really amazing data set to start teasing apart some of these rules in more detail. But for today's purposes, I just wanted to give you a small vignette about how we're approaching this using incubation experiments and really feeding it back into those time series observations. Before I show you any actual data, I wanted to provide some acknowledgements here. A lot of different people have touched on these data sets over the last years, but I wanted to highlight current group member Hannah Anderson, who helped out with some assemblies, and she and Haley, who did a lot of RNA extractions, and then former group member Matt Harkey, who also spent time working on various data sets that have informed this talk. And last here, I'll highlight Harriet Alexander. She and I really started our first metatranscriptome experiment focused on the eukaryotes using incubations in this system. And those experiments and ideas have filtered through our thinking about how the system works. So we're going to be tracking responses to resources in the keystone phytoplankton, again, asking who's there and what are they doing? Just to get down into some nitty gritty here for this audience, we typically take roughly 20-liter samples per replicate. You can see this is a major filtering operation to try to do it fast, and at a decent flow rate. We collect filters that often look blank, which is always a little disconcerting, but have worked out for us. And then in this case, I'm highlighting the fact that we actually took each sample and did 2 different library preps. The samples were collected onto a 5-micron size fraction, and to really focus on those larger eukaryotic phytoplankton groups. And we did unselected libraries to, in part, look at the expressed taxonomic diversity of the community composition. And we did greater than 5-micron poly-A selected libraries to look at the express functions. This is our Meta-eukomics workflow. So again, we took both libraries, quality control, the ribosomal signals. We're using a program called RiboTagger, and then we used assembly-based approach inspired in part by the eurythmic workflow that Ariana Krinos has put together. So this includes assembly, sequence alignment, taxonomic annotation using ukulele, and functional annotation using eggnog, and then generating our assembly product and looking at orthologs, differential gene expression, and other signals. One novel aspect of the signals that I'm showing you is that we normalize to a set of stably expressed reference genes from our incubation. So these are taxonomic signals where we've normalized the functional gene count to a reference set, and I'd be happy to answer questions about that at the end. All right, so our focus was to simulate resource addition. So what is the influence of vertical mixing that I showed in that diagram of the biological pump? And to do this, we did some initial experiments. The very first ones I did with Harriet Alexander, where we said, okay, let's take deep sea water and add it to surface water, and then look at the in situ sample, the control, and the deep sea water amendment, which I just called DSW for short. And then we've done this experiment multiple times. Here's what the community structure looks like after that experiment. And you can see that we have an increase in the relative abundance of diatoms in deep seawater amendment. So deep seawater addition, mimicking a potential vertical mixing event, sees an increase in chlorophyll, which is why the DSW treatment is green, and a shift in the relative abundance of diatom signals. One of the striking features of this style of experiment is that every time we do it, we see a similar response. So here's a set of experiments from 2015, another set from 2012. And although the details may vary in terms of the magnitude, you can see there's a relative increase in diatoms every time you add deep seawater to the community. What's even more striking to me is if you go in and ask, well, what diatoms are responding? It is one of the same two taxa every time. Every time we do this experiment, the community consistently reorganizes, and the winners are Cylindrotheca and Pseudonychia. You can see that here in the rank abundance plots where the in situ is on the left and the deep seawater amendment is on the right. And I've just color coded in the triangles and squares, the Cylindrotheca, which is the square, which is the winning diatom in all of these incubations. So again, year over year, we get a consistent response. Now, this begs the question, we have this reproducible response to deep seawater, but what is it about deep seawater that is driving or underpinning this response? So for that, we move to semi-factorial treatments. And here's an example of an experiment that we did to identify what the resource driver is in that scenario. So we took deep seawater, nitrogen, and a treatment where we added nitrogen, silica, iron, B12, everything but phosphorus, which we call the no P or the minus P here in this picture. And then we did the same thing for a no nitrogen and added phosphorus. And you can see here in the green color that all the treatments that get any kind of nitrogen, you see a chlorophyll response. And in fact, if you look at the distribution of taxa, again, you can see an expansion of the diatoms in any treatment that got nitrogen. This is perhaps not as dramatic for nitrogen addition alone, there in the middle. And so, although nitrogen is a key driver of shifts in the relative community composition, there do appear to be synergistic effects from other resources. Now, another way of viewing this is to go in and ask, well, in these treatments, did we see a Cylindrotheca response in the plus N and the minus P? And in fact, we do. Here's the relative proportion of expressed ribosomal signals for diatoms and haptophytes. You can see there's an expansion of Cylindrotheca in green and to a lesser extent Pseudonychia in the treatments that got nitrogen. That's plus N, minus P, and DSW. So the winning diatom, again, is consistent across all treatments with nitrogen. We can then go in and ask about what expression profiles, what genes are underpinning this response, and how do the expressed functions really map onto this. And here I have expression profiles plotted in a correspondence analysis. And if you just look at the clustering, those triangles are the in situ. In purple here, we have all the treatments. Again, this is a profile of all expressed Cylindrotheca orthologs, and you see that minus P, plus N, and DSW are all clustered together and distinct from any treatment that didn't get nitrogen. Now, I promised that we were exploring drivers in situ, and I want to go back now and take this incubation and put it in context. So just as a reminder, here's our shipboard amendment samples, again, showing the expansion of Cylindrotheca and the community composition. And I want to show the in situ samples that we have to go with this. So these are samples that span July and August. They're near Station Aloha in the NPSG. These are the same sample. So this is the in situ for our incubation experiment. And if you look at those 2 p.m. in situ samples, again, what was kind of amazing is that we seem to have captured an in situ expansion of Cylindrotheca relative abundance, which happened in August. So, of course, the question immediately comes up, what is underpinning this shift in relative abundance of Cylindrotheca. And here we delve down into the expression patterns, which vary over time. So here in the triangles, you can see in blue and green, we have the in situ, and then you move through time. And again, what's striking is that when we look at express functions, the nitrogen addition treatments and those August samples cluster together. This really underscores that nitrogen may underpin the observed shifts in expression patterns and community composition for that expansion that we see in Cylindrotheca. In my mind, this is learning the rules. One final bit of data before I wrap up, and that is we went into those expression patterns and asked, are there variations in nitrogen signals that might underpin this and help reinforce that this is a nitrogen-driven signal as opposed to something else? And that's what you see here. These are nitrogen metabolism-related genes in a heat map with the normalized gene expression plotted. So this cluster analysis, you can see that the samples in July cluster away from the other major cluster, which includes the incubation samples and the August samples. So these are nitrogen functions, and you can see that the unregulated functions are consistent with a role of nitrogen in driving these in situ dynamics. We also looked at phosphorus, and the signal for a sodium phosphate cotransporter is here. And these signals suggest that this P-regulated transport, which is going up in plus N and minus P, which did not get phosphorus, are not up in any of the in situ samples. So again, phosphorus doesn't appear to be a driver here. So just to wrap up, we've shown that nitrogen is a key driver of these keystone phytoplankton physiological ecology and the concomitant ecosystem functions for this system. And what I hope I'm leaving you with is that by tracking these non-steady state dynamics, we can help identify ecosystem structure function relationships and try to understand the rules in which the system is operating. In this case, nitrogen stimulated, which really could be simulated vertical mixing, is a consistent driver of increased phytoplankton abundance, and phytoplankton community restructuring happens in favor of diatoms. As I mentioned, we're working on further resolving this dynamic with monthly hot samples pending. And I'll just wrap up there with my acknowledgement slide and open it up for questions if there's still time.
Thank you, Sonia. That was fantastic. Yes, thank you so much. I have some questions, but let's open it. I would love to open it up for a couple of questions before we move on. I can also start, and you can submit questions on the via the chat in Zoom, or you can also go ahead and raise your hand or just unmute yourself because we're a good friendly bunch. One, yeah, let's start with Natalie here.
Hi, Sonia. That was a really beautiful talk. Thank you for that. I thought that those trends with Cylindrotheca were really interesting, and we're seeing that it's also a responsive diatom in shelf communities. But I'm also interested in the dinoflagellates. That seemed like they were relatively abundant, which I know we have to take with a grain of salt because they have very large genomes. But has your group looked into which dinoflagellates appear to be relatively abundant out there, because it seems like they're doing quite well in a system where there is no nitrogen, and just kind of on the flip side of what you were showing, yeah, could there be some interesting physiology there that allows them to exist in that environment?
Yes, absolutely. I was trying to pull up a slide, which I'm not going to be able to do, just to cross-check myself, because we have looked at least what taxa they are. I don't want to misspeak, so I'll have to catch you later on that. But they are mainly heterotrophic or mixotrophic taxa, and so I think a lot of that dinoflagellate signal could be related to grazing, and we do see some interesting dynamics in the context of the known heterotrophic dynamics and boom-bust cycles in potential prey items. We've looked at this more for diazotrophs in terms of thinking about grazing on diazotrophs, which of course also play a keystone role in the system. But I think there's a whole story to be told about the dinoflagellates out there that we really haven't sunk our teeth into yet, but would be such an exciting area of further inquiry.
Thank you.
Yeah, and we have time to later after both speakers, we can continue discussion, show other slides. We have time for like one short question where while Lucia maybe gets ready for putting on their talk. Anyone else? I do have a short one, and it's I don't know if there's an answer, but I'm excited about this monthly hot time series. Based on I don't know if your group has had a chance to look at the nutrient data, but you know the semi-factorial experiments and then monthly time series. Do you think that there's opportunity to compare across months or years to look for that same increase in nitrogen resources, or do you think you'll capture some of that like you have in your experiments, or be able to relate it to your experiments?
Yeah, well, I'm not sure that we would be able to resolve that in terms of the nutrient dynamics. I mean, one of the beautiful things about the metatranscriptomes is that it allows you to query the organisms themselves for that physiological signal. And I haven't looked at the nutrient data in terms of nitrogen across the sample set that I just showed you real recently. So, but it is very typical that we see no difference in nitrogen across there, right? We wouldn't predict it from the nutrient dynamics because that is just taken up so fast, and it doesn't reflect the competition and the interactions that are really underpinning how the system works. And that's where I think looking directly at the physiology helps provide those clues, and that the experiments and looking at other types of forcing, for example, across eddies or over the diel cycle, can help to identify regulation patterns and how different kinds of forcing influence that. I'm also super excited about delving into the time series and thinking more about how we mine that in the context of the work that has and continues to go on in that system.
Great, thank you. Lucia, do you want to go ahead and get started soon? So thank you so much again, Sonya. Put my notes here. So Lucia is tuning in from Naples, and so we'll be mindful of time too. And so Lucia, thank you for joining us. She's a staff scientist at and she's going to be presenting some work from a large group project and how they're using multi-omic methods and other approaches for a large project that's set in the Western Mediterranean in the Gulf of Naples. Please go ahead and
thank you very much. So I share my screen.
Yes.
Okay. You see it?
Looks perfect.
Okay. Thank you very much. I'm Lucia Campese, and I work at the Stazione Zoologica Anton, Dohrn, Naples, Italy. And first of all, I really want to thank you for inviting me for this seminar. It was already a pleasure to meet some of you in Buzzol in 2023. And I'm very happy to to be able today to talk about the main project I involved in, which is Nerea, which, as Sarah mentioned, it's a project, is an initiative that aims to integrate multi-omics analysis for marine observation in Naples. So the long-term topic is actually shared with the talk from Sonya. And of course, we are also interested in marine observations and environmental omics to understand the ocean functioning. And Nerea is based on this mainly. And as in Bioscape, so our efforts are based on the interdisciplinary connections between different fields, omics approaches, biochemistry, and modeling. Nerea stays for Naples Ecological Research for Augmented Observatory, and has three main objectives. So on one side, we want to advance the long-term research on marine ecosystem dynamics, but we also want to focus on specific phenomena and specific processes through targeted field studies, like in the case of the impact of rivers on the sea, for example. And we also want to establish a continuous crosstalk between field observations and laboratory experiments. The study site is very different from the North Pacific subtropical gyre that was mentioned before. It's as Sarah mentioned before, it's the Western Mediterranean Sea, so the Tyrrhenian Sea. Here, I hope you can see my pointer, but here there is the city of Naples, and Nerea mainly sample on three different sites. One where the sampling is monthly is the long-term ecological research Marichiara station, which is 2 miles off the city of Naples, and it was established in 1984. It's one of the world's longest ecological time series focused on plankton and also the chemical state of the water column. We also sample in two other sites in the Gulf of Naples. One is the Sarno River mouth, where Sarno is one of the most polluted rivers in Europe. And another site is the canyon Dohrn, which is a submarine structure, which is also a biodiversity hotspot. So we covered three different sites in the Gulf of Naples. The Sarno River is sampled seasonally, and the canyon Dohrn 2 times per year. And we do monthly sampling at the Marichiara station. The main sampling strategy and workflow is very similar to what was done for Tara Oceans, which inspired us to start Nerea. And we do a size fractionation sampling. So we collected samples from filters with different mesh sites, and we do collect different sites of organisms, and we also do use nets for collection of phytoplankton and zooplankton. So different size fraction and different approaches to be able to really cover all the ranges of sizes of planktonic organisms. And we started the processing of samples at sea through seawater filtration. So we filtered the water through different progressively smaller mesh size filters, so 20 microns, 3 microns, 0.2. And we actually also collect the viruses at the very bottom of this process. And so this is just a picture of how this is installed for real on the boat. So we have a peristaltic pump and the 3 different filters. Once the filters are collected, we immediately froze them in liquid nitrogen, and then we go in the lab, and there will be the extraction of DNA and RNA and the sequencing for metagenomics, metabarcoding, and metatranscriptomics, and also the extraction of proteins and metabolites for liquid chromatography and mass spectrometry. as also Sonya mentioned, we ask the very basic question. So we want to know taxonomic information of who is there. So we extract DNA to sequence metabarcoding and metagenomics. So both the 18S, 16S metabarcoding markers for amplicon sequencing, and we'll do also shotgun metagenomics to understand which are the species that are there, and also to understand which is the metabolic potential of the species that are present, so what they can do, which are the set of genes they are equipped with for dealing with the environment. But also we want to study what is the actual metabolic activity of the species that the communities that we find. So we are also sequencing RNA for metatranscriptomics, and we are also collecting and processing proteomics and metabolomics. The project Nerea started in 2019. So it's the Marikara station, the long-term ecological research started in 1984. But then since 2019, Nerea aimed to augment also the sampling of the long-term ecological research by adding to the physical parameters, the nutrients, the microscopy counts, the flow cytometry, all the other omics, which I told you about. So we are still sampling. There are some holes here and there in the sampling because of several reasons, mainly here with the pandemic and so on. And of course, the status of the data is very different because some data are easier to process, some are less easier to process in terms of cost and timing and computational power and so on. What we recently managed to publish was and to release were the first 10 months of data. So from April 2019 to January 2020, where we published and released the data for all the physics and all the nutrients, the microscopy counts, and prokaryotic metabarcoding and eukaryotic metabarcoding and metagenomics of prokaryotes. So for the prokaryotic metagenomics, we actually relied on the collaboration of the group of Shinichi Sunagawa in Zurich, where they went through single assemblies of our samples to generate in parallel a functionally characterized prokaryotic gene catalog and almost 4,000 of metagenome assembled genomes, so MAGs. And so we now have a collection of genes in the catalog that can be either linked to MAGs or to assemblies. This is what we're also trying to do with eukaryotes. And these data are not published yet because with eukaryotes is a little bit more challenging, as I think many of you know already. So what we are doing is in parallel the metagenome assembled genomes reconstruction and annotation to build what is actually a catalog of Nerea MAGs, and the meta-T assembly to generate a catalog of Nerea eukaryotic genes. So I'm gonna go a little bit in some details of this pipeline here. So for eukaryotes, we did a co-assembly of the 60 samples from the first 10 months. I say 60 because it's different size fractions, sometimes different depths. And then we generate contigs, mapped back the reads to the contigs, and through we generated statistics on contigs like K-mer frequency, differential coverage. And we applied a semi-automatic binning. So first we clustered contigs with automatic binning, and then we ended up with a set of meta bins or clusters, which are basically different MAGs in the same group of similar things based on K-mer frequency and different coverage. And then we did the manual extraction of and curation of MAGs from the meta bins. And this very last part was done by Antonio Longo, PhD student in our group, where he went through this is just a picture of an example of an eukaryotic MAG that we extracted. So this is the interface of the software that we use, the Ambio, where each bar here is a contig, and each horizontal line is a sample, and they are clustered here according to sequence composition and differential coverage through Euclidean distance and the word algorithm. And this cluster that you see here, which is a MAG, it is something that when you click on it, just simply on the interface, it gives you some statistics on this, what is supposed to be a genome, so a MAG in this case, which in this case is a very nice, let's say, MAG with a high completion, a small redundancy value. And it's taxonomically assignable to the diatom Pseudonychia, which was already mentioned before. And so we did this kind of analysis for all the meta beans that were generated with the previous pipeline. And we ended up having 52 eukaryotic MAGs corresponding to these first 10 months of Nerea. What we're dealing with now is the gene prediction and annotation of MAGs, also including the metatranscriptomic data of the corresponding samples for that. And of course, the aim at the end will be to integrate all the omics data that we have to study the taxonomic profiling of samples and species turnover, the microbial community functioning in time, and the chemical interactions among the species and environment. And this wouldn't be able if we didn't have many collaborations with the different institutions. I already mentioned the group of Shinichi Sunagawa in Zurich, but there is also Genoscope in France, and the group of Max Aito for the metaproteomics, and Georg Ponerty in Germany for the metabolomics. In parallel, we are also studying the abundance expression patterns of target species and enzymes that were already, for which we already have reference genomes, transcriptomes, and proteomes. Together with this main overview of what we are going to do with the long-term part, let's say, of Nerea, we are also trying to focus on specific processes through targeted field studies. And this is the case of a Lagrangian process study that we performed last year in April-May, where we aim to unravel the physical, chemical, and biological response of river influence from shore to sea. And this project was in the context of a bigger project, which I think maybe some of you already know about, which is TREC, Traversing European Coastlines, which was a project that aims to study and investigate what happens from in the continuum from the sea, from the land to the sea. And in this context, TREC was able to traverse all the coastlines of Europe, and Naples was one of the super sites where the truck with the lab, the mobile lab, stopped, and the researchers stayed more, and we could be able to actually perform this experiment. So this is just the chance that we had to have the truck with this mobile lab in our park where the Stazione Zoologica is. So we still didn't have all the sequencing done for this experiment, but what we did was we followed the river output to characterize the chemical, the physical, and the biological responses to the riverine input from the onshore injection to the offshore dissipation. And to do that, we also did that with a simultaneous sampling with different boats. So there was a Stazione Zoologica boat and the Tara schooner and another boat to sample in the same moment different part, let's say, of the filament. So more inshore and more offshore. And yes, and another aim was, of course, to close the carbon budget, including the air-sea exchange, and relate everything to the process studied in the previous aim. Finally, one thing that we aim to do, and we are starting to do with the NEREA, is also to link field observations with the lab experiment, which is something that is very important to do when you collect so many data from sequencing. And to do that, we just started a project, a PhD project to study diatom bloom dynamics in the Gulf of Naples, using all the metabarcoding, metagenomics, metatranscriptomics, and metabolomics data, and especially focusing on the domoic acid production in the model diatom Pseudonychia multistriata. Pseudonychia, that was already mentioned in the previous talk, is actually a blooming species in the Gulf of Naples. Pseudonychia multistriata in particular is one of the many species of this genus which is able to produce this toxin, this neurotoxin, domoic acid, and we actually already found some signal of the presence of this neurotoxin through the metabolomic data. So what we're doing now with the new PhD student is to correlate these cycles that we identified in the metabolomic data with the expression of domoic acid genes and the presence of potentially toxic diatoms. So again here to correlate to the previous talk, which I really liked, this is also a way that we thought to actually study the rules of the system through this crosstalk between lab and the field observations. So yes, to finish, I just finished with some final consideration and challenges, and I hope to engage in some discussions later. So I think we all agree that a multidisciplinary and integrative approach is essential to address the complexity of marine ecosystem. And NEREA, in this case, given the history of the long-term ecological research and the, let's say, small scale, geographical scale of the sampling, can serve as a case study to test hypotheses, methods, and models, then be applied to larger scales. And of course, this comes with some challenges. Some of them are not really solvable in a short moment of time, but I try to list some of them to maybe also trigger some discussion, let's say, about that. So we have the issues that all omics methods are database dependent, and lots of data are actually lost during the pipeline from the sampling and sequencing. Also, we have the challenge in obtaining absolute values from sequencing data, and this is something that also came a lot as a discussion in the Bioscapes meeting of November 2023, about also the integration of omics currencies in models. And there is still high cost of some sequencings, especially for the metatranscriptomics, for example, and the high computational demand for bioinformatics and downstream data analysis. And as some of these analyses are new, some of these technologies are new and always improving, there is also a lack of standard protocols and pipelines. And in this context, NEREA is also part of an ongoing effort to develop bioinformatic pipelines for the analysis of metagenomics and metatranscriptomics data with a focus of eukaryotes. We are building a working group co-led by me and Federico Barbals from Buenos Aires for the analysis of this kind of data from Mission Microbiome Atlanteco, which is another project focused on the Atlantic Ocean. We are collaborating with Genoscope and the École Normale in Paris for really put it together the efforts. And of course, I think Bioscapes, in particular, this group is really for me a great chance to put all the efforts together on that. And I think I'm done. Thank you very much. I would like to thank all the people involved in area, which are really a lot. And thank you all for your attention.
Awesome. Thank you so much, Lucia. That was great. It sounds like a fantastic project. So many samples being processed and to be processed, and it's a great body of work. And the challenges at the end, the bottlenecks, definitely resonate with this community for sure. So lots of stuff there. I had I'm going to open it up to questions, but I had one quick question. I was curious if you can share any insight. I was especially the Lagrangian study that you shared. That looks like a really neat data set with the influence of the river output there that you're following. Do you have a hypothesis, or do you have any you follow the Pseudonychia bloom, but are there other community, like major dominant members of the phytoplankton community or microplankton community that are kind of signaling where the river input is, and then trails offshore more?
Yeah, well, for the Pseudonychia blooming, we know that it's a regular blooming diatoms in the Gulf of Naples. It's normally end of summer and beginning of October. But the thing is that it's also changing a lot in the very few last years, let's say. So it's also a species that you can find in colder waters and also with the higher temperatures. So one of the we don't have a signal for so far they didn't identify any specific environmental trigger for that. I was actually thinking about the deep sea water thing, because we for example in the long-term Marikara station we sample at 0 meters and in 40 meters for the majority of the year, except in winter, because in winter it's very mixed, and so we just sample the 0 meters. But as far as I know, there is no specific environmental trigger like nutrients for that. But this is clearly something to investigate.
Mm-hmm. That's great, thank you. So I'd like to open it up for targeted questions, especially related to this project. Yeah, so folks can enter questions on the chat, or they can unmute themselves. And maybe, Lucia, while your slides are still shared, if you don't mind going back to the bottleneck and challenges page. I feel like that's a good discussion.
They're still there. Okay.
Yeah.
Yes. Yeah. You see it?
Yes.
Okay.
Yeah, so I think that the Meta-eukomics group met previously, and we're continuing to meet, and some of the challenges and bottlenecks are definitely on the top of the list. But can you elaborate a little bit more on the database dependent? Because I know you talked about the gene catalog. I'm familiar with like the methods to generate that, and then I know that there's at least one Terra Oceans transect through the Mediterranean. And so when you talk about database dependent, what are you talking about, like a reference database, or like using
yes.
that gene catalog and other? Okay.
Yeah, I mean, it was more a general observation about how you I mean, just the amount of unknowns that you obtain every time you sequence anything from the environment, like half of that or something, it's unknown, and everything that you can find is just similar to what was already published. It's just a really trivial point. It's just sometimes we forget. I try to keep it in mind because it's always. I mean, I always find it I don't know, challenging to claim each time that we describe an environment, and then we also know that there's so much that we don't know yet, and this is something just a point of just consideration.
Yeah, I think I had an entire like almost 2-hour meeting yesterday talking about, are you sure it's okay that this much data loss for my metatranscriptome stuff is okay? And it was just a reassurance thing.
Yeah.
Maybe others can chime in about that.
Lucia, I really enjoyed your talk. Thank
Hi.
you for those overviews that were really well described. I wanted to ask you about the databases, and in our working group, we talk a lot about how these methodological decisions can really change the products that you end up with. And you were talking how important that is for your inner comparisons just within your expeditions. So you're working with Genoscope, which has been a part of Tara, and you've worked on these other projects. So was there internal dialogue to figure out, okay, this is the database we're going to use to be consistent with these other studies? Or is it more of, this seems to be the best, most robust database, so we're going to switch and use this different method going forward? Can you talk us through like what that decision process was like with your team?
Well, it's much more confused than that because the thing is that there's always something new. So of course you want to update everything with the newest database, and or you want to build the in-house stuff. Like for example, we also have some reference genomes and transcriptomes from species that were isolated and cultivated from the Gulf of Naples. So for example, we like to have some groups really working on these species since ages, and they want to keep these references for studying something that is trackable in the Gulf of Naples. But for example, we had a discussion in December on the use of Marferet, for example, for the annotation of our mags. So yes, this is really something that it's not decided yet. It's really an ongoing discussion. Yeah.
Makes sense. Thank you.
Yeah.
Anyone else? And Lucia, just as one of our previous webinars, the writer of Marferet spoke as well. So I think there might be some information about that too, if you have a follow-up.
Okay, thanks for
Sure.
Okay.
Everyone's just reminded about loss of data with metatranscriptomes. Is that what? Yeah. But yeah, I really appreciate you chatting with us today. Yeah, does anyone else have any questions for either speaker, for the group? Can I ask one more question, Lucia? Because you guys all have a great sampling schematic. Can you maybe discuss or what about training, like for bioinformatics training? Like
Sorry, what
about the training of the computational demands? Like your last a bottleneck here is computational demands of bioinformatics and downstream data analysis.
Yeah.
And I wanted to ask about training folks on how to do that work, and I was curious.
Yeah. Well, what we're planning to do with the Mission Microbiome Atlantico data is actually to gather some people in Naples in next spring, to have a look at the data that are done, that are ready from some of the legs of the project. And well, I don't know. I think I don't even feel myself as an expert in bioinformatics, but I learned how to, for example, extract the mags and do all the pipeline from the Meta-G to the mags. And I think that the key will be to, well, the aim of this training, let's say, of what we're going, this working group, is not only to train like technically the bioinformatics, the scripts or the pipelines, but also to think at what kind of question you can ask to this kind of data, and which is the, it's more on the conceptually speaking part, and then for the bioinformatics, it's always evolving. So it's I don't know, really. I hope it will be good.
No, I think it's something that's always evolving, which makes it definitely very challenging to keep up with. That's something I'm always learning. So I was hoping you had like perfect advice for me to figure it out, but I'm just kidding.
I would love to. Yeah.
So you don't have to share your screen anymore if you don't want to, but
Okay.
we'll open it up for a few more minutes if anyone has questions for Lucia or Sonya. Otherwise, we'll give them a big thank you for joining us today. And as always, you can learn more about the OCB Meta-eukomics working group. Like I said, we're a friendly group, and so again, I feel like it's a great group to contact if you have a question about meta-transcriptome, especially eukaryotic ones, data interpretation or database issues or something. And so, yeah.
Well, thank you very much, anyway, for the invitation.
So
and
much.
Also, Sonia, thanks for your talk. It was really inspiring in these 20 minutes.
Yeah,
nice.
thank you both so much for coming and sharing your science. And we'll all talk to you soon.
Thank you for having us. It was a pleasure.
Thank you, Heather.